Hi, welcome to this pre-practical introduction video. My name is Ludovica Griffanti and I will give you an overview of the third and last practical on task FMRI. As usual, we have different sections and I will cover the content of the core sections, but there are optional extensions if you are interested in the topics. The first section is about motion and physiological noise correction, and we will see how to apply the techniques that we saw in the advanced pre-processing lecture. We will look at all but the ICA-based cleanup because it will be covered in the resting state ICA practical. For this section, we have 2 repetitions of the pyramids and palm trees task, the same that we used in the last practical, that were acquired in the same subject, but in one scan, the subject deliberately moved and breathed irregularly. So we have what we called nice and naughty dataset. So first of all, you will be asked to look at the report of the analysis on these 2 scans and see what is the difference. So one thing to look at when you look at the reports side by side is the pre-stats tab, where you see the plots of the motion correction. So at first glance, it seems that both subjects moved quite a lot, but actually you need to keep in mind that the y-axis is scaled differently. So you will see that actually in the nice scan, the subject moved very, very little, while in the naughty dataset, the subject really moved a lot. We now try to correct the data from the naughty scan. First of all, we do simple motion correction. So starting from the design matrix that we set up in our basic analysis, we add the motion parameters. And this is the design matrix that we have once we add the motion confounds. And you will be asked to look at the results and see what's the difference. A second option is to use PNM. So we will give PNM as input the recordings of the cardiac and respiratory signals. And in this particular case, the data were acquired with multi-band acquisition. So the slice order will need to be specified in a separate file. One thing to keep in mind is that if you don't see the txt file in the list when you click the button, it's because you probably need to delete the word image in the filter so that PNM will show you also the known images and you will be able to click on the txt file. Once you run PNM, the first thing that you get is to see the cardiac and respiratory trace, and you can check that PNM detected the peaks correctly. You can then run the next command that is reported either at the bottom of the page or in the text of the practical. And this is the command that will actually create the EVs, the voxel-wise EVs, to then add in the design matrix. And again, we ran some analysis for you, one only including PNM regressors, and the second one using both the PNM regressors and the motion confounds, so combining the first 2 options. And again, you will be asked to look at how things change in terms of activations. Finally, we look at FSL motion outliers, so we want to try and remove the effect of sudden motion. You run the command, which generates a txt file with the outliers, and because we use the verbose option, you will get some more information appearing on the terminal, and you will see where FSL motion outliers found spikes that you can check in Fossilize to see whether you agree that there was a sudden motion in the correspondence of those volumes. Finally, we can try and apply all these techniques together. So you can set it up in feet. There is the drop-down menu for adding motion parameters. We need to add PNM confounds as voxel-wise confound, and you can add the motion outliers using the add additional confound EVs and giving the txt file as input. And again, you will be asked to see what's the effect on the final activation. The second section is contrast in parametric designs. And the example that we will look at is very similar to the one that was presented in the lecture. So in the lecture, you saw an example of auditory word presentation at different rates. Here what we have is reading sentences presented one word at a time at frequencies ranging from 50 words per minute to 1,250 words per minute. Very similar to the lecture. First of all, you are asked to look at some pre-calculated results, and in particular to look at the first 4 contrasts that we set up and to the F contrast. So as you can see in the design, we have 1 EV per frequency. We used temporal derivatives, and the contrasts that we have are for the differences between different frequencies. You are then asked to think about what the F contrast means. So what are we looking at when we set up the F contrast? If you don't know or don't remember, you can check in the lecture, or there is some hidden text in the practical that you can check. Once you looked at the F contrast, we need to think about what is the shape of the response. And to do this, we are going to look at the parameter estimates for the main EVs. So we combine the PE images in one 4D file with FSL merge, and we look at it in Fossilize. So after a bit of setting up, like described in the text, you will end up with the results of the F-test, shown in red, for example, in this case in red-yellow. And the plot at the bottom will show you for each voxel what is the value of the parameter estimate for the 5 PE, for the 5 regressors. And if you click around the brain, you will see that in some areas we can see an inverted U shape of the response. So first of all, you are asked to think about why that could be, and then we now want to test these more formally. So we need to think about how to set up our contrast to test for this shape. And this is when you are asked to look at the contrast from number 5 to number 8, where we set up a linear and quadratic response. And then you are asked to think about which one do you think is the one that is best to model and to investigate the inverted U shape that we see. So I'm not going to reveal that, but when you find it, you will load it in Fossilize, and you will see that, in fact, the voxels that are significant for that contrast are, in fact, the ones that showed an inverted U shape. The next section is about interactions, and for these we will look at a dataset where the subject was performing a motor task, a visual task, or sometimes both. So the basic analysis that we set up is by modeling only the motor and the visual stimuli separately, but what we are asked to do is to set up a third EV which models the interaction. And you will see that, in fact, the interaction term corresponds to when the motor and the visual stimulus were both on. And you will see at the results at the single subject level and also at the group level. And the last section is about contrast masking. And for this section, we're going to use data from the word generation and word shadowing task that we used in the first practical. In particular, now we are going to look at the differential contrast, number 4, so shadowing greater than generation, because as it is, the contrast doesn't tell us whether the areas that are significant are because shadowing was more positive than generation, or because generation was more negative than shadowing. So this differential contrast doesn't tell us anything about the sign of the activation. So what we need to do to investigate that is to set up a contrast masking. So in particular, we want to see where in the brain there is a significant difference between shadowing and generation, so where shadowing is greater than generation, but also when generation and shadowing are positive. And to do this, we set up a contrast masking, masking contrast 4 with C1 and C2, and you will see how the results change doing this. And with this, I thank you for listening, and if you have any question, just ask the tutors.